Okay, I think you can see my slides, hopefully.
Looks good.
All right. So yes, I'm also presenting on some work done evolving Syn3A, similar to Roy. This is work I did when I was still a member of the Bernard Paulson's lab at UC San Diego. So a lot of my PhD was focused around using evolution as a tool, and we called it ALE, Adaptive Laboratory Evolution. You just leverage the fact that you keep a collection of cells growing and propagating, and natural selection will naturally select for the things that are best, and those will win out in the population just from serial propagation of these cultures. And so if you can generate these fitness trajectories along the course of evolution where you have a starting strain that grows at a certain growth rate or whatever your fitness metric is, and then as evolution progresses, you get these jumps in fitness as beneficial mutants sweep to fixation in the population, and you can get multiple rounds of this, and then you go in and you sequence the DNA of these organisms, and that will let you know what are the causal mutations that give you this improved phenotype they've developed. So you're sort of essentially Monte Carlo sampling genomic sequence space and just letting this population of millions to billions of competing microbes tell you which mutations are best by seeing what wins out. In the Paulson lab, we have ALE machines, robotic systems that help us run these evolutions in a throughput and with data quality you couldn't get manually. So we do it in large flasks of 15 mil volume. It's a robotic liquid handler with custom software, handles things, and it goes in and samples tubes and will serially propagate while keeping them at a set temperature and tracking fitness over time. So in this case, we took the Syn3A strain and we evolved it through this ALE process. So we did 10 independent replicates. Starting from Syn3A, we evolved to SP4 knockout growth medium at 37 Celsius, and we evolved for about 400 generations, maintaining exponential growth phase through serial propagation. Generally, when we run ALE experiments like this, we get a nice fitness trajectory where we can actually see the jumps in fitness as beneficial mutants fix. Syn3 is a little harder to measure with optical density readings in that way, and so we weren't able to get a real-time fitness trajectory, although improvements to the automated systems make it so that we could potentially, through different wavelength measurements, get that type of trajectory in subsequent experiments. So when you look at the phenotypic change after evolution, you see that pretty consistently across our evolved strains, they decreased their doubling time by about 15% compared to the Syn3A ancestor. So there is room for fitness improvements, and they're pretty consistent across the strains. There were no wild outliers that got much better or much worse than the other strains. They seem to be converging to the same sort of phenotypic state, and that's something you see in evolutionary experiments where those first mutations you acquire in those first sweeps are generally the most important for fitness, and then you get diminishing returns as more mutations accrue, and you sort of asymptote to a relatively stable, evolved growth rate. So there's no endpoint for evolution. This could be kept going indefinitely, but you'd get decreasing returns as evolution progressed. We saw some morphological changes in the evolved strains. So in two of the independent lineages, we observed mixed populations that developed where upon plating, you would see both large and small colonies that seemed to be coexisting in the culture together. This was sort of consistent over the course of evolution, where even at midpoints, we could see this arising, 200 generations in, and then they were still around at 400 generations. This is something that is seen in evolution experiments sometimes if there are different niches that the organisms can be living in, and so they're sort of coexisting by occupying different niches, or if they've evolved some sort of syntrophy where they rely upon the presence of the other to serve some metabolic need that they've purged. The fact that they have about the same growth rate when measured independently, not cultured with the other, indicates that these are actually just competing mutants that are at roughly the same phenotype, and given sufficient time, you would see one of these win out. And so that shows sort of, even with the slow asymptoting towards improved fitness, there's still room for improvement, and further sweeps would be possible if evolution were kept going. When you look at, so we did whole genome sequencing on all these evolved strains, and if you look at the mutations they get, you see strong signatures for the fact that adaptive evolution is taking place, not simply neutral drift. You see much more non-synonymous than synonymous mutations. The types of genes that are targeted fall into this categories we regularly see mutated in these types of evolution experiments. Again, because we're sort of mainly selecting for exponential phase growth rate, you see a lot of transport reactions that get upregulated because flux into the cell can be rate limiting, and also increases in translation and transcription and everything that lets the cell grow faster. And we saw very distinct mutational trends that distinguish SYN3 from E. coli, which is what we normally evolve in the lab, where almost all of the SNPs caused an increase in the AT percentage of the genome, and only 2 actually led to a G or C being added. And of all the insertions or deletions we saw, almost all of them were deletions, and there was only one base pair insertion. So this just shows that SYN3 has a very distinct mutational trend compared to other organisms, but not necessarily different than mycoplasma itself. And so then we actually looked at all the mutations, and I know there's a lot on this slide, but this is what I love to see after an evolution experiment, where there's not a ton of mutations in the endpoint strains. The end of 400 generations, they only had about 8 mutations per strain. So that makes interpreting the results much easier than if they were hypermutable and had dozens to hundreds of mutations, which we see sometimes. Clearly, among this small subset, some of these are important, if not all of them. And you also see quite a bit of reproducibility in the genes that are targeted. So these ones that are called out in colors are genes that independently across these strains acquired multiple distinct mutations. And that is a hallmark of the genes most strongly under selection. So in that same way where as you get these fixes in beneficial mutations that are sort of rank ordered in how important they are for fitness, this is sort of a proxy for that if you don't have that time series DNA sequencing data where things most frequently hit are most likely to be improving fitness statistically. So let's dig into a couple of examples of these genes frequently targeted for selection. TET-M is in SYN3A. It was not grown in these conditions with antibiotics present. And so it turns out that having to express the TET-M with whatever promoter, ribosome binding site is naturally in SYN3A is energetically wasteful enough that multiple strains chose to downregulate this through either intergenic mutations or premature stop codons or frame shifts. So this makes sense. This confers no benefit and is just wasteful when you don't have antibiotics around, but it also shows that its expression is actually impactful enough to the cell that it's one of the strongest targets in a mutation where you only get, or an evolution where you only get like 8 mutations by the end. This is an example of an uncharacterized efflux ABC transporter permease that got 5 distinct amino acid changing mutations within it. This is a model of what it should look like roughly based on some computational software and where the mutations we see fall. And so this is, unlike with TET-M likely being deactivated, this shows clear signs of altering the function of this transporter. It could be upregulating, increasing its flux capacity, perhaps, or it could be changing the substrates that it acts upon. This mutational list provides good resources to go and make mutants to test sort of what these changes are doing and characterize the genes. Another pretty consistent change we saw across a number of strains was this collapse in TA repeat number that falls upstream of an uncharacterized lipoprotein, where in the wild type SYN3A, it has 13 of these TA repeats, and multiple strains independently decrease this number down to 9, 10, or 11. This is a form of phase variation where, unlike SNPs or indels, these slip strand mispairings that cause expansions or contractions of these repeats occur at a much greater frequency mutationally. And so strains are able to find them more quickly. And so it's 1, easier to get, and 2, if it provides a selective advantage, then that's going to win out in the population. And we saw through some RNA sequencing that this did increase expression of the lipoprotein. And another thing I found quite interesting was several, uncharacterized proteins, the genes still in SYN3 that we're not really sure what they're doing, were nevertheless repeated hits of mutations across strains. This top example, it changed the start codon or it caused a premature stop codon. So clearly it seems to be deactivating that protein. Whatever this protein is, it's not good for fitness under these growth conditions, at least in the context of the other mutations in these couple strains that got it. Whereas this uncharacterized protein, it seems to be changing its function through non-synonymous SNPs. And so this shows how, even if we're sort of in the dark a bit on everything that's going on in the cell, we can look to evolution to point us towards things that are most strongly affecting selection. We also did RNA sequencing on these strains, and we saw trends that make a lot of sense in the context of other ale experiments. These were strains evolved to increase their exponential phase growth rate, and we see upregulation of ribosomal proteins and downregulation of repair proteins. This is the classic greed versus fear trade-off. They get greedier because they're not living in stressful times of hitting stationary phase or running out of nutrients. And so they just upregulate everything that makes them grow faster. And are sort of lose their fear. Whereas if you evolve them to fluctuating environments or low nutrients, their selective pressure would be different, and you would see a different trend than this. And they fell into sort of two main clusters that just looking at the mutations the strains acquired doesn't really make clear why they delineate in this way. That's something that metabolic modeling and more single mutant characterization could help to contextualize. You can also see TET-M is here, and it has this one strain, AL5, that has a much, much lower expression, and that matches perfectly with the TET-M mutations we see where some actually change the protein with a frame shift or a stop codon, but this AL5 strain got two upstream mutations that we deduced were downregulating expression, and sure enough, that is what they are doing. So overall, I was happy to see that SYN3A is suitable for evolutionary interrogation in the lab. It doesn't just acquire a ton of mutations and its genome's so unstable you can't figure out what's going on. It reliably achieves fitness improvements of double-digit percentages, and the mutations make sense. They agree across strains, even if we can't perfectly explain what's going on. And so there's a lot of future work that could be done where using ale to find these mutations and then characterizing them to uncover gene function, using metabolic modeling to better understand what's going on transcriptionally in the cells. And you can, of course, do evolution to all sorts of different environments, maybe more stressful ones, maybe ones with coexisting or infection-based, do it with different genetic engineerings in maybe the SYN3B strain, and with better fitness tracking. So ideally, you could get actual real-time fitness trajectories and know when exactly a mutation had swept into the population. So with that, I will take any questions.
Right. Can you remind me, how were you measuring fitness exactly?
Doubling time. So just doing a growth curve and measuring double-stranded DNA over time and getting cell number from that.
Awesome. Thank you.
What would you see as a logical follow-up to your experiments?
I think the gene targets are the clearest follow-ups here to dig in deeper to what specifically those mutations are doing. This shows that at least under these growth conditions, these are the most important genes to change to improve fitness. And so if these growth conditions are useful to understand better, or the gene targets themselves are useful to understand, like perhaps the uncharacterized ones, this sort of serves as a great avenue in which to explore them because we know they have an impact on fitness here. Or I would say if there are particularly interesting growth conditions or starting strains that are maybe somewhat different from SYN3A or tweaked in the selective pressures they're under, just sort of throw a bunch of different replicates in a tube and let them grow for a while and see what mutations they get. And you don't need robots to do it. It just helps. But that's easy enough to do, and whole genome sequencing is very quick now. So I think this could help inform models better and also just get gene function.
One thing I'll note, one of your slides, you had the growth rate of SYN 3.0, at 3 hours. And while that's what we published in 2016, we took 2 clones of Syn 3.0, and one had essentially the same, but not exactly the same sequence. One divided every 3 hours, the other divided every 120 minutes. So, that's a slight correction, but it might be useful knowledge for someone.
Good to know.
Okay. Thank you, Troy.